Hello, my fellow Everthinkers. Today we're joined perhaps by the most eminent hospital pharmacist at our university, Professor Daan Touw, specializing in therapeutic drug monitoring and toxicology. Professor Touw has contributed to both fields as a researcher and clinician for over 40 years. Today we will explore therapeutic drug monitoring and various techniques associated with it, along with developments in toxicology. And Zed, what did you think was the most interesting thing we discussed today?
Reflecting back on the episode was really one of the broadest episodes that we have covered. A lot of research topics have been covered, a lot of topics, a lot of new ideas. I was intrigued by the prospects and about the new horizons that could be discovered in the future. So, really recommend the episode.
Yeah, and I guess Daan Touw really showed us how multidisciplinary he is as a human.
He's from the old generation.
Exactly.
He doesn't want to focus on one thing. He has a problem at his clinic. He sees a problem at the hospital, and he wants to go back to his lab and focus on it. So,
exactly.
That's how real science works, in my opinion.
Exactly. And as you can imagine, many problems arise, so the interests are different or let's just say are varied every single week. So it's a fantastic podcast with a lot of different concepts covered. So really take your time, listen to it, and thank you so much for listening.
Yeah.
Professor Daan Touw, thank you so much for joining us on this a little dangerous and wet morning. In your own words, could you introduce yourself to the audience so that they have a better idea of who you are?
Thank you for inviting me in the first place. Apologies, I have a little bit of a cold. Why did I study pharmacy? Because it's a very broad study with mathematics, physics, chemistry, biology, pharmacology. It's in my view, it's one of the broadest scientific studies.
Very good.
That's why I chose it.
And maybe give us a small background of your career. Where did you start? In what period of your life did pharmacy kind of inspire you to pursue it, and where did it take you?
Okay, I was already inspired when I was 16, so that's a long time ago, nearly 50 years ago. And why was I inspired? One of my uncles also was a pharmacist, and I saw what he was doing. It interested me, and then I chose to study pharmacy in Leiden. At that time, you still had a pharmacy in Leiden, the classical education. And after 7 years, I first had to go into the military service. That was obligatory at that time yet. And during the military service, I have to go one step back. During my study, I did several interns to see what I liked most, both in a community pharmacy and in a hospital pharmacy. And there I noticed that hospital pharmacy was more interesting, broader, more scientifically than the community pharmacy. And during my stay in the army, I investigated which hospital pharmacies had vacancies, and then I applied for a job. At that time, there was no formal training to become a hospital pharmacist yet, as it is now. It was more informal. And so I applied at several hospital pharmacies, and in the end, I went to The Hague to work in the hospital pharmacy.
And was the idea of research always present? Was it something that you always want to do, or was there a specific moment that you said that this is the moment that's inspired me to do research or to go in the direction of research?
When I went to The Hague, I didn't. No, that's not quite the case. When I did my internship in Sittard, that's in the southern of the Netherlands, near Maastricht, I was doing research, and I liked research. So when I went to The Hague, I already knew that research would be my main interest. And of course, you have to work, you have to do patient care, but I wouldn't say from my first day, but already very early in my stay in The Hague, in my career there, I started with research projects. And that was, for that time, quite unusual.
Mm-hmm. And so you first did your hospital pharmacy residency, and then you went on to do your PhD.
Yes, I first did my hospital pharmacy residency in The Hague. That took 4 years. And then a pulmonary physician went from Leiden to The Hague together with his patients. Those were all cystic fibrosis patients. And cystic fibrosis was kind of a myth that time. You didn't know what the disease was. We thought that patients were pharmacokinetically different from normal, not cystic fibrosis patients, and I decided to study that. And he liked it, that I wanted to study that, and the two of us, Wim Bakker, that's the pulmonary physician, and I went on studying. We started with the pharmacokinetics of tobramycin in cystic fibrosis patients.
Interesting, because from now what I understand is that your main drive to research the cystic fibrosis pharmacokinetics was basically the disease itself. But then you pivoted basically for the rest of your research to go into pharmacokinetics, mainly. Yeah. So not into cystic fibrosis drugs or something like that.
Yes, the disease and the pharmacokinetic behavior in the patients. They were malnourished at that time. The disease intrigued me, and the effect of the disease on the pharmacokinetics. That's true, yeah.
Mm-hmm. And was the field of pharmacokinetics at that point similar to the field of pharmacokinetics now, or what was the main difference in your time at this time?
At that time, you didn't have all kind of modeling software we have now. It was merely hand calculations. We had we had the first personal computer in 1986. So
1986.
That's so before that, the first personal computer came. You had to do it by a hand calculator or by hand.
In what year did you do your PhD?
I did my PhD in 96, but I started with the studies for my PhD in 88, 89.
88, 89. So the personal computer was just coming to...
You had to write your own software in DOS or in other basic computer languages. I'm not a computer scientist, so I'm just a simple pharmacist. And you had to learn how to operate in disk operating systems and not in Windows. Windows didn't exist yet, and programs were not plug and play. You had to define your own software.
Exactly. I can hardly imagine how it was without like the internet and the personal computer that we know today. Because now if I want something, I just type in something in PubMed and it gives me hundreds of information and hundreds of information sources. How was it that day?
PubMed didn't exist.
It didn't exist.
We had books, thick books, and in those books they were in those books were listed all papers. So you had a book and you were looking at cystic fibrosis, and you get a list of all papers on cystic fibrosis in the past 1 or 2 or 3 months. So that book was issued every 1, 2 or 3 months. And you didn't have that many journals as we have now. Perhaps you had perhaps 100 journals, and of which 5 or 10 were interesting enough for me. And you didn't have hundreds of papers per day. You had only 1 or 2 papers per day that were published.
Wow. And so I can imagine that in your time you have seen the field exponentially developing.
Yeah,
from basically papers and few papers. Yeah.
From doing everything by hand to what you're doing now, computerized automatically. I've seen the whole development, yes.
So to quickly cast back a little bit, because the role of a hospital pharmacist may be ambiguous to ones who haven't been exposed to it, or to ones who don't have experience from a pharmacy background, because it's usually behind the scenes. Could you give us a small, I guess, TLDR of what a hospital pharmacist is, what a toxicologist does, that sort of thing at a hospital?
Yeah, hospital pharmacy in Netherlands is different from abroad. In abroad, you are usually only involved with the distribution of drugs, getting the right drug to the right patient at the right moment. In the Netherlands, we have a large tradition of compounding our own drugs. So at that time, we rather compounded our own drugs than bought them on the market. Nowadays, it's the other way around. When a drug is on the market, you buy it, and you do not make it yourself. But that day, we had a large tradition of compounding our own drugs. So we made tablets, we made capsules, we made injections, infusions, millions of tablets per year. We had machines doing that. And the pharmacy where I was working in The Hague also had a war function. So as a kind of backup for the military pharmacy, it was at the end of the Cold War. You needed to have facilities when a war should break out, that Holland could make his own drugs. So the pharmacy in The Hague had such a function. And nowadays it doesn't have that function anymore, but when I joined it, it still had the war function.
Okay, very good.
And you mentioned already toxicology. So the pharmacy labs are very well equipped with good equipment. You can analyze drugs, not only in the compounded drugs or in the raw materials, but also in human samples, in blood, in urine, in tissue. And from history, the Dutch hospital pharmacists also had a large role in toxicology. Usually, toxicological investigations are performed by forensics labs, but in the Netherlands, it was performed by hospital labs. And the Dutch Forensics Institute only dealt with criminal cases, but all other toxicological cases were investigated in hospitals.
Okay, very good. And how does the balance between these two roles work, I guess? Because, you could say that they are slightly different. One is to treat the patient and to kind of see from, for example, a disease work towards making the patient better. Another is, for example, perhaps where someone was poisoned by something or overdosed with something. How do both roles differ? And is it a logical kind of step to kind of be very specialized in both?
Due to the size of the task, you needed to specialize. During your traineeship, you were educated in all fields. But, for example, when dealing with a production pharmacy, when dealing with a production area, with people working there, that is a full-time job. And not only for one person, but that time for two persons. Dealing with the laboratory of the pharmacy is also a full-time job for two persons. Dealing with the logistics in the pharmacy is also a full-time job. So already in way back in the 80s and the 90s, we had to choose, because the hospital pharmacy in The Hague was a large pharmacy. It was responsible for the pharmaceutical care in the whole city of The Hague. So we had 6-7 hospitals at that time in The Hague. We had nursing homes. So tens of thousands of patients you had to take care of. So that made you already specialize in an early phase. In smaller hospitals, you performed those tasks of multiple tasks within one person, but not at a level as when you can do it when you're specialized. Mm-hmm. So it's largely different whether you are working in a large facility or in a small facility. In a large facility, we already had an early specialization.
Okay. Interesting. And also you're working in an academic facility. And one of the things that I personally am always impressed by is that you are a professor, but you are also a clinician. How do you manage to balance research with your clinical work?
In my opinion, you cannot do research without clinical work, without both your legs in the mud and seeing what's actually happening. I'm not a scientist who is in an ivory tower. I want to know what's happening in real life, and I also want to deal with real life problems and try to solve them. And then I go back to my first research. For example, the cystic fibrosis research, I saw a problem, a clinical problem, and I want to solve that. And then I want to see if it works. So in my opinion, as a medical scientist, you should be on both sides, both in the clinical side and in the research side. But I also know that not all my colleagues agree with me.
Yeah, well, this is the essence of science. You look at something in practice and you want to solve it in the lab.
Yeah, that's my view.
Exactly. But what I always wonder is that professorship is already 4 jobs together. You have to manage things, you have to teach, you have to ask for grants, you have to research, and on top of that, you also take care of the patients and of the patient cases. How do you manage to balance these tasks together?
I think I have a lot of experience because I have 40 years of experience now. And in those 40 years, you learn how to manage more tasks within one job. And I'm only a professor for 10 years now, so I can fall back on a lot of clinical and practical experience. So I do not have to invest a lot in getting that experience. I am sure that a younger colleague who gets all those tasks at the same time might have more problems in managing it. Mm-hmm. But yeah, I think it's experience, which is the magic word.
Interesting. So it's one of the tasks gets easy, and then you could basically squeeze more work parallel to that task.
Yeah.
That's very interesting. And of course, I can imagine it's also passion, that also you have a lot of passion for research. It drives you, it gives you more time, it gives you more energy.
If you don't like it, you won't, you won't survive such a job, I'm afraid.
I think so, yes. Talking about research, we usually start with the research topics immediately, but this time we thought we'd change it up a little bit. We want to ask you, what is the most interesting research that you think you're doing at the moment, or the research that you are really interested in?
I have a lot of interests. That's my problem. But my most intriguing project now is the toxicity of the alpha-amanitin containing mushrooms like the death cap mushroom and the victims it makes every year. And now we are trying to elucidate how the antidotes work, because based on history, some drugs are given as antidote, and we really do not know how they work. And we also want to find out. We want to develop a method to measure the toxin in human materials, so you know you treat the right patient, because the antidote is not very easy to obtain.
Mm-hmm. Interesting. And how does such a toxicological research work? So basically, what is the main lab work in this toxicological research?
The main lab work consists of a few parts. We have an analytical part in the development of an analytical method. Our basic technique is mass spectrometry, and we're trying to develop a mass spectrometry method. But we also have experience in developing ELISAs. So with having antibodies against this toxin, we already have developed an ELISA for research purposes. And other lab work focuses on the toxic mechanism of the toxin, and we look at toxicity of the toxic effects on the liver, the toxic effects on the kidney, and the toxic effects on the bone marrow, because you see liver damage, you see kidney damage, and you see a reduction of the production of blood cells.
Mm-hmm. And do you exactly focus on the molecular mechanism of these pathways, or is it more on the organ level?
We morely focus on the organ level because we usually use organ slices, or in the case of bone marrow cells from the blood.
Okay. And you said that there is some ambiguity regarding antidotes for especially some of these toxins that historically we've kind of accepted, okay, this antidote has worked in the past. Well, why is that? Is there a difference in pharmacological pathways that kind of causes this confusion as to why one antidote may work or why it shouldn't work but maybe does? How is that?
Yeah, we still don't understand the mechanism. We think we know how the toxin works. It inhibits the RNA synthesis by inhibiting that pathway. And therefore, the toxin has to enter the cell. And we know that patients usually come too late. They eat the wrong mushrooms, and the next morning they have abdominal pain. But the toxin has a very short half-life, so the toxin has already done its work. The majority of the toxin has already been removed from the body by the urine because it's eliminated very quickly. So kind of hit and run effect. And I do not understand why we usually start the antidote 24 hours after the poisoning, and still the antidote work. So the damage is going on, and the antidotes are supposed to work by blocking the entrance of the cell by the toxin, but the toxin has already been there. Yeah. It hasn't been a hit-and-run effect. So why do the antidotes still work? And that's what we try to find out.
Okay. So is it more like, could it be also that the antidote has a antagonist role in some receptors that the toxin already?
We don't know. It also might be the scavenging of oxygen radicals due to the damage of the cells. We don't know.
And so we don't also know anything about the mechanism of the antidotes from other research or something like that?
No, there's very limited research. And what has been demonstrated is that the antidotes inhibit the entrance of the toxin. But what I just told, the toxin has already done its work. So why do they still work while the toxin has already for the major part left the body? That's intriguing me.
Interesting. And how long have you been doing this research?
Oh, we're working on it for a year, about 4 years now. And the problem is that no commercial company is interested in funding this. So we do it with master students, master projects, and students doing this for a short internship for 2 months.
So is it not? Is the clinical problem not widespread that people are interested in it, or is it something that's recently coming up?
It's a typical Dutch problem because the mushroom is very common in the Netherlands, but in the northwestern countries of Europe, we are rather mycophobic in the Netherlands. So not many Dutch people eat these mushrooms, but refugees from mycophilic countries who are used to pick mushrooms and to boil their food with it, that's the majority of the victims.
Interesting.
So it's not a big issue because Dutch people, I guess, have grown out of scavenging mushrooms, which makes sense.
We all learn at school never to pick mushroom from the bush because there's one which is extremely toxic, and the risk you die is enormously.
Makes sense. And maybe we can actually stick to toxicology now that we've actually moved into it. You mentioned the fact that analytical techniques are used, of course, and you did say that mass spec in particular, I guess, is used for biomarkers specifically. Are there any other techniques, or ELISA, of course? Is there anything else that is required, for example, for testing for illegal recreational drugs? Or I read a paper recently of that you were involved in regarding e-liquid constituents and their toxicity levels, perhaps. So yeah, any other techniques that are perhaps used?
No, our techniques presently are all based on mass spectrometry, because the new developments in mass spectrometry, the equipment is so extremely sensitive in the microgram or submicrogram per liter region that this is the preferred technique. In the past, we used ultraviolet detection or visible light detection or electrochemistry, but mass spectrometry is far more sensitive than the other techniques there are.
So the other techniques have kind of been phased out.
They are
very rarely
they are
used
phased
nowadays.
out.
Okay.
We still have them, but we prefer mass spectrometry.
And we even heard from you recently about the epidemic of designer recreational drugs and how the fact that they're reaching the Netherlands, being consumed by people here, and then causing various health risks.
Yeah.
What is the problem with, I guess, identifying these drugs and dealing with them from a clinical background? What issues do you run into regarding the raw drug itself, its metabolites? How are you able to even identify that it's a new designer drug that this person has been poisoned by?
Designer drugs are designed to mimic the effect of existing illegal drugs like amphetamines, benzodiazepines, opiates. And because the opium law is a restrictive law which identifies the compounds that are illegal. So to avoid the law, clever chemists do large modifications on the molecule to have a kind of legal amphetamine, a legal opiate, a legal benzodiazepine. So because the purpose is to mimic the effects of benzodiazepines, opiates, and amphetamines, we know how to treat the patient as if it were a classical opiate, benzodiazepine, or amphetamine. So for the treatment part, we know what to do. But for the analytical part, it's difficult because you do not know which compound someone has taken. And with a normal mass spectrometric method, you must know at which mass you should have to look at. But there exists equipment that is able to screen also not yet identified compounds, high-resolution MS, Orbitrap equipment, and with large international libraries, online libraries, and when you have the exact mass, you can make a guess which compound it is. But somebody must have identified it already somewhere in the world.
So you basically get a library, or you get, when you screen, you get basically a lot of signals from all the drugs that are in the blood, and then you have to know exactly which compound are in these signals?
Yeah, you need to have very powerful software to screen all those signals. It's not only drugs in the blood, but it's also waste material from the body, amino acids, and other body-own compounds. You need very powerful software which tries to identify which mass you have found, and then a combination of peaks brings you in the direction of the probable compound, because the compound is fragmented. So you see the parent and you see fragments, and the combination of those masses lead you in a certain direction.
But you have to know which compound you're looking for in the first place.
No, not with this machine and not with this software. This software can really think. It's kind of artificial intelligence. It combines signals and comes with a probability.
Oh, probability that this is the compound that's causing...
Which compound it is, yeah.
But I still have difficulty imagining it because there are hundreds of compounds in the blood. How do you know that this is the compound that you're looking for, or like?
Yes, but it's a combination of several properties of the compound. First, you use HPLC to separate all compounds in the blood, and based on the retention time, you have a lipophilic or heterophilic drug. You have a kind of distinction in which direction you have to look, and further, you identify a peak and you get an exact mass. And having the same molecular formula but a different structure gives a different mass. It's not a mass in mass units, but in thousands or 10,000s of mass units. So every chemical structure, although having the same molecular formula, can have a different mass.
Interesting. And you mentioned that there is a growing role of AI in all of this.
Yeah.
Do you see a lot of research going into the direction of AI and this screening methods?
I think this screening method already has a lot of AI by combining these signals, but perhaps with AI you can even get more powerful and more quicker your results.
And is there something that we're doing in the department regarding AI?
We had someone in our department specifically on artificial intelligence, but I think at that time we were too early. And now we are rethinking it again.
So, to move back to, I guess, the role of a hospital pharmacist and how we really understand what is happening in the body, we have to, I think, backtrack towards therapeutic drug monitoring. What is therapeutic drug monitoring in its most simple sense?
Therapeutic drug monitoring is using the analysis of the concentration of the drug in human material, usually plasma, to adjust the dose of a drug and thereby improving the outcome of a drug. And it sounds simple, taking a plasma sample and measuring the concentration of a drug, but there is a lot of pharmacokinetic modeling behind it. And so we highly rely on pharmacokinetic models, simulations, and also studies towards the outcomes between the concentration of a drug and the clinical effect.
Exactly. So basically, the premise of therapeutic drug monitoring is there should be a relationship between the efficacy, toxicity, and the blood concentration.
Yes, the concentration alone is not predictive enough. It's the combination with kinetics, dynamics, and in the end, the clinical effect.
Mm-hmm. And why is it so difficult or why is it not so easy to find the relationship between the blood concentration and the other parameters?
The most simple answer is that the pharmaceutical industry is not interested in this relationship. They want to give a flat dose. One dose, one size fits all for someone who's 40 kilo and someone who's 120 kilo. Perhaps a little bit adjusted to body weight, but not scientifically adjusted.
So shouldn't it be obligatory or something that's necessary to do by the pharmaceutical company that they should, for example, know that this efficacy or this drug concentration is the optimal drug concentration before releasing this drug to the market?
I would like that, but the regulators do not yet. In the past, they even didn't ask for pharmacokinetic studies. Nowadays, they are obligatory, and we can also use pharmacokinetic studies to build the pharmacokinetic models, but it's not yet obligatory to prove that there is a relationship between concentration and effect and use that knowledge to improve or to optimize the dose. Because when you prove that, you also must provide an assay. And drug companies are not analytical companies. They are companies who produce drugs and not analytical assays. So they must find a partner to develop an analytical assay.
Exactly. So you basically have first to know how to extract the drug from the blood before knowing the relationship between the drug and the blood concentration.
Yeah.
I was thinking about therapeutic drug monitoring when putting these questions, and one of the questions that popped in my mind is that why don't we go exactly to the target? For example, the intracellular concentration. Why do we go to the blood?
I would love to go to the target. But, for example, an antidepressant works somewhere in the brain.
Mm-hmm.
You don't drill a hole in someone's skull and take a sample of the brain to measure if the concentration of the antidepressant is okay. What I hope to establish before I retire is mass spec imaging. You know what imaging is. You look at at labeled drugs, usually radioactively labeled or fluorescent labeled, with a optical method or a radioactive method. Look with a gamma camera, for example, to radioactively labeled drugs, and then you can see with the three-dimensional camera where in the body the drug is. But you have to administer radioactively labeled drugs to the patient. What I hope we can establish here in Groningen is that you can take a little bit a sample of tissue out and look at and slice it and look at it with mass spectrometry, liberating the drug with a laser beam, catching it in a mass spectrometer and measuring the local concentration of the drug in a piece of tissue on a cellular scale.
And what's the added value of this method?
Then you know where the drug really is, and then you can establish the relationship between the local concentration in, for example, a tumor for a oncological drug and the blood concentration. Then you can relate them to each other when you simultaneously take two samples.
Interesting. So if I understand it correctly, we take a biopsy from the patient, a piece of tissue, and then we basically localize where the drug is, and then we measure. We use the mass spectrometry to know the amount of the drug in that specific area.
In that area, in that cell.
That's very interesting. And what's the main limitation, or what's the main barrier at this point for this method to be established?
Money. This equipment is costly. You need equipment to liberate the drug, matrix-assisted laser desorption techniques, and you need to have a special mass spectrometer that's able to catch the liberated molecules and to analyze them. So it's a costly hobby.
And is it time efficient? Time-wise, is it time efficient to do that?
It's not time efficient, I think. It helps us to better make a relationship between the local concentration at the target site and the concentration in the blood. Because the concentration in the blood is very easy to measure in all patients, but you cannot take biopsies in all patients and not from all tissues. So in selected patients, you can, and there you can study that relationship, and you can use that knowledge perhaps in other patients.
Oh, interesting. So we basically use this method not for everyday practice. We use it basically to establish a relationship between the blood and the target concentration, so as research tool. And then we could basically use the blood concentration as we do it usually for TDM, but then with more precision.
Yeah, you can use it as a research tool, but for example, for local tumors, for skin tumors, you can also use it for beyond the research tool, and you can also use it for daily practice. But first, you have to develop the technique, of course.
Mm-hmm. And I'm thinking now sometimes there's no relationship or at the population level between the blood concentration and the efficacy and the toxicity of the drug. But then when you use this method, there should be a relationship always, right? Because you're always measuring at the target.
Yeah, I think without the drug at the target, there is no effect. So there must be some relationship at least. Yeah. And I want to know from how many drug is on target, how many drug is off target, causing side effects, and how can you optimally dose that as much as on target and less off target and causing side effects.
And do you need to go at a molecular level to know what is bound and what is not bound to the receptors, or is
that would be ideal? But then your resolution is so extremely high. The present techniques are on a cellular basis and not on a receptor basis yet. But perhaps in the future, you can also do it on a receptor basis.
Interesting. But maybe a parallel idea, since we have, for example, tumors in the or cancer in the blood for T cells or any other immune cells or white cells, would it be an idea, for example, to go and measure the blood or the concentration of the drug intracellularly in these cells? And the drug, isn't that more easier than, for example, in the brain or any other organ?
Technically, it's possible. You can separate the cells and you can measure it, but you must know whether the relationship or the concentrations you measure are related to the concentrations at the target of interest. So that's the first step you always have to make.
But isn't it more precise than measuring the concentration in the blood as a whole?
It depends. It depends on all kinds of transporter mechanisms that are involved in and exporting the drug. I think that for every drug it has to be investigated whether a plasma concentration or a cellular concentration gives the best reflection of the concentration at the site of at the target site. But you also have to keep in mind that measuring cellular concentrations is not very easy and not a technique every laboratory very easily can perform. Plasma concentrations is rather easy, but cellular concentration, you have to separate the cells, you have to select the right cells with a kind of methodology, and then lyse the cells and measure it, or try to measure it in a single cell. But it requires, I think, quite some experience,
exactly, yeah,
and some work.
Mm-hmm. We talked about TDM being used in tumors and cancer, so I think this is a good moment to talk about why TDM is very important in cancer therapies, tyrosine kinase inhibitors therapies.
TDM is important because it helps us in optimizing the dose with increasing efficacy and reducing toxicity. And especially in cancers with the new oral anti-cancer drugs, it's a huge development. Nearly every month a new drug is licensed. We have more than 60 or 80 at this moment developed within about 10-15 years. And in the past, we were used to dose until the patient didn't tolerate the dose anymore, so the maximum tolerable dose was usually administered. But for the new oral drugs, where cancer kind of becomes a chronic disease, let's hope, which you can suppress, the approach should be different, because you have to live long taking the drugs, and you cannot dose until the patient doesn't tolerate the dose anymore. So you have to more balance between efficacy and toxicity, that you have maximum efficacy, but also for the duration of the treatment with limited toxicity. That means that we have to dose differently, and then it might help if there is a relationship between the concentration in the plasma and the effect of the drug, and then you can target to the right concentration range.
And how well would you say that at this point the therapeutic drug monitoring is being used for TKs? Is it well established at this point?
It's an emerging field. The Dutch Pharmacology Oncology Group, that's a group of Dutch pharmacologically interested oncologists and pharmacists working together. And for some TKIs, it's now established which concentration gives maximum effect with minimum toxicity, and that field is that number is growing.
Interesting. So for the TKIs or any other drug, you need the patient to be at the clinic. You need the patient to be in the hospital to basically take the sample and give it to the lab technician to basically do the work. But me and Oscar were also intrigued about one of the techniques you were also busy with. It's the dried blood spot. Could you maybe tell us something about how this facilitates the whole clinical approach to this?
Yes, the dried blood spot technique is already an old technique. You know it from the heel prick from newborn babies, but it's qualitative. Having an inborn error or not. For quantitative measurements, it started in neurology, in patients treated with anti-epileptics in the 90s of the previous century. And when I was working in The Hague in the cystic fibrosis field, I developed an assay for tobramycin in dry blood spots because The Hague has a regional function or a national function. We had CF patients from the whole country coming to The Hague. And for their drug monitoring, we developed a dry blood spot technique so they could take a sample at home, and it's a very simple technique. You do a little prick in your finger and 1 drop of blood on a piece of paper. You let it dry and you send it by post to the laboratory, and there the sample is analyzed. So it gave these CF patients the opportunity to stay at home and not making long journeys to the hospital just for a blood sample to look if the drug concentration is correct. And the same you can also do for TKIs and other drugs. And the major important thing is that the drug should be stable in a dried form, in a dried blood spot on a piece of paper. And that must be investigated very thoroughly before you can use such a technique.
Are there any drugs that at this point we use therapeutic drug monitoring with DBS, with dried blood spot samples?
We use it in psychiatry and in transplantation. And we have developed it for the immunosuppressant drugs before the COVID period, and it was very helpful that we had this technique during the COVID period when patients couldn't travel, couldn't go to the hospital. You had to reduce the amount of outpatient clinic visits. So we were glad we had a dry blood spot technique so patients could draw the samples at home and send them to us. So we got a massive increase in the dry blood spots during the COVID pandemic. But for TKIs, we haven't developed it, but I know that the National Cancer Center in Amsterdam has developed it for a few TKIs, but merely for research purposes. So patients can draw multiple samples at home and send them to the Dutch Cancer Institute, where they are analyzed. And I do not know if they also are using it for routine drug monitoring.
Interesting. And you also mentioned COVID. Me and Oscar were also discussing this. A lot of research we see from other professors that a lot of research has been accelerated during COVID. Was this dried blood spot samples research accelerated after COVID?
No, we had developed it before COVID, so we were glad we had the technology in place to maximally use it during the COVID period.
Mm-hmm. What do you think the effect of COVID was on the research in general?
Depends on it's because labs had to be shut down for a part. You had to work in teams, a morning team, an evening team, an afternoon team, or a week 1 team or a week 2 team. So you could do less lab work. But on the other hand, many reviews have been performed. So perhaps many many more background knowledge has been collected instead of work from lab experiments.
Interesting. So because there was less time in practical work, there was more shift towards the background meta-analysis, statistical work, research work. That's interesting.
And perhaps that has also led to less duplicate of work when you do more in-depth your background investigations. But you can never compare it with the...
Yeah, that's an interesting idea, though.
But it might be interesting.
Mm-hmm. And moving on to maybe another aspect of the hospital pharmacy, TDM as well, partially. Pharmacokinetic modeling itself. It's, to my understanding, it's very critical to reduce, for example, animal testing, or when you don't have human material, it is also very useful. Could you give a small explanation as to how it works, what it's based on, and maybe how you use it on the daily basis?
The classical way of pharmacokinetic modeling is based on multiple samples drawn in an animal or a human, and then using mathematical techniques to build a 1, 2, 3 compartment model with drug input, drug input, output, drug distribution, several compartments. But due to the more powerful computers, we are shifting towards physiologically based pharmacokinetic modeling. So you know what the liver does, you know what the kidney does, you know what muscle does, you know the blood flow through those organs. You can put that already in a model, and then with less samples, less animals, less humans, you can already create your theoretical model and do sparse sampling and sparse and having only a sparse number of volunteers or animals to validate your model. So PBPK modeling, that's the abbreviation of physiologically based pharmacokinetic modeling, goes more in the direction of a validation of a theoretical model than really building a model.
Okay.
So in the end, it should save lab work.
Yeah.
Animals.
Okay,
very good. And I guess
one of the questions that we also were discussing this. You want first to do a good lab work to know what the organ is doing and have good information to basically put it back in the system to predict the lab work again. So there's a circular relationship in a way.
You're completely right. You have to start with measurements. There's a Dutch famous saying, meten is weten. And without measurement, you have no knowledge.
No knowledge, exactly. But then, so for example, these models at this point, are they good enough to predict the real life models that we see in animals or slices or humans?
They are increasingly becoming better. And we have, for example, one of my colleagues has built a PBPK model of a pregnant woman with cystic fibrosis and treated with a combination of the potentiators of the CFTR enzyme. And we had the opportunity in one patient to measure on a regular basis some levels, and it nicely fit the model. So with there's only one example, but I think the software and the basic models that are already there are very good in predicting. You only have to tweak them with samples, but with less samples as what you should have needed to create a model.
So there are, to my mind, almost zero limitations to this technique. It's you create the model based on a little bit of clinical experience. You then perfect it using extra clinical knowledge and extra clinical experience. And then in the end, it should be a very kind of nice predictive tool for a specific subpopulation that might be using a specific drug, for example.
Yes, that's nicely summarized.
Very good. Okay. So, yeah, that's amazing that we're able to, I guess, make it with such limited data, kind of make this almost model that works every single time. And yeah, I guess with very few anomalies, I assume.
Yeah, but I'm also thinking that the predictability is always limited to what we know to build the model with.
Yeah,
So
that's also important. You know the limitations of your population you have built and validated this model with. When you, for example, go to severely overweight patients who have a totally different disease, you know that the model has to be adjusted, and perhaps in the future you know which buttons you have to turn to adjust the model.
Yeah.
What the excess of fat, for example, has the influence on your pharmacokinetics. For some drugs we know, but for some drugs we do not know yet because we have no experience with those drugs in, for example, the overweight patients.
So at our department, is the research more into developing these models, or is it more into modeling the pharmacokinetics of some drugs?
We do both.
Both, okay.
Both, yeah.
And is there any specific kind of drugs that we are actually working now with?
No, we are working with the tyrosine kinase inhibitors. We are working with the CFTR potentiators in cystic fibrosis. There are colleagues working on anti-diabetic drugs. So we cover a few fields. And antibiotics, of course.
And you mentioned earlier the case of pregnancy. I also heard that we also have focused on the pregnancy modeling and the placenta modeling, but also from another side, from the organ on a chip modeling. Can you maybe tell us something about both organ on a chip and the pregnancy situation?
Doing studies in pregnant people is it's not ethical and it's difficult. So we are looking but we want to know what the safety is of drugs for the unborn child, and usually drugs are avoided in pregnant people, but sometimes they need it, and then you want to know the risk for the child. And a method can be a placenta on a chip or sliced placentas after birth of a child. But the problem is that after birth, the placenta usually is grown, and you also want to know in earlier phases of the pregnancy how drugs migrate over the placenta or are metabolized in the placenta, what transporters are there, and then the organ on a chip technology might give an answer, but that's still in its very infancy.
What do you see? You say it might. So do you see a more optimistic hope that it would be?
I hope so. Okay. I hope so. Yes,
I think. See more limitations?
Of course, there are limitations because you have to create an environment, an artificial environment, where a placenta develops until, for example, 10 weeks old or 20 weeks or 30 weeks. How do you create that environment? Because the human body is so complex. How do you manage that? And I don't know yet. A colleague of me is working on it. But let's say I'm very hopeful. But we also have to realize that it might be much more complex than we think at this point.
Yeah, because you need to create an environment so you don't only need the placenta, but also a placenta and other also cells that also create some or signals.
Other cells, but also compounds circulating in the blood. hormones or other compounds that influence the development of a placenta.
And do we think at this point that we have the sufficient background information to know what influences the placenta so that we could model it in an organ-on-a-chip model?
I think we are one of the forerunners worldwide. The colleague who's working on this point has contact with other research groups in the world. So we are not there yet. It's a long way to go, but it's a very intriguing road she is going.
Mm-hmm. Interesting. And if I imagine it correctly, we also need the placenta to grow, or do we need to model the placenta and an organ-on-a-chip at a specific trimester or a specific point of time?
You can do both, but the problem with organs on a chip is that their longevity, the length that you can keep them well is only limited. So you have to think, do I do a kind of longitudinal study, or do I create artificial placentas which have a specific age? I don't know what the way is to go, but at this moment, an organ on a chip, it has a very short lifetime.
Well, I assume the limitation would arise if we were to stick to placenta models that we design for a specific week or trimester. The limitation would come that drugs that affect, for example, the vasculogenesis of a placenta, for example, could cause certain issues in certain areas. And so you wouldn't be able to balance that. So you'd hope to, I guess, recreate the real life scenario as faithfully as you possibly can, which is very interesting. So it's not just about getting the right transporters in the right places and kind of going, okay, we've got everything that is on a human placenta to our knowledge. We need to, well, yeah.
I'm also wondering, do we have the knowledge?
Exactly.
Do we know all the cells and all the compounds that need to be put in the model so that we have a good model?
Exactly.
Do we have, by the way, a good in silico model for the pregnancy, or do we still also struggle with that?
There are PBPK models for pregnancy, but the transfer of the drug to the unborn child is very difficult to predict. There are some methods to calculate, to estimate it by the pK and the pH of the mother's blood and the child's blood, the fat solubility. It's very difficult to predict because there are also transporters, importers and exporters, which interfere with passive processes. From passive processes, you can try to predict, but the transporters which interfere with the passive processes are less well to predict.
And so what is now our best modeling tool for pregnancy? Is it, I don't know, is it actually ethical, or is it allowed to have pregnant animals to test on?
Nowadays, I don't think you have to look at the regulators. What do the regulators want? That is investigated. You can simply say, my drugs are forbidden in pregnancy. Then I think that the regulators are okay, that you do not do any tests on animals or whatever. But if you allow your drug during pregnancy, the regulator will ask you to demonstrate that the drug is safe during pregnancy. And that's difficult. Because perhaps the child will be born, but what's the development after 30 years in a child that is exposed to the drug and versus a child that's not exposed to a drug?
Yeah, and we don't want a catastrophe about what is the name of the drug? Yeah, exactly. Yeah, it was a catastrophe, and probably we don't want to repeat that. So there's more strict regulation at this point for that.
Of course.
Yeah. There is always a question that we ask at the end. What is you as a researcher, what is the $1 million question? What is the question that you think that, okay, I want the next generation of researchers to research this and to put their effort and focus onto this question?
Oh, I have a lot of
I can imagine.
A lot of wishes. Yeah. I have not one $1 million question. I have a lot. I hope that the alpha-amanitin problem will be solved. I hope that we have a good placenta on a chip for my other colleague. And I hope that we are able to measure, for example, TKIs on a cellular basis in a tumor. Those are my 3 $1 million questions. And I have a lot more, but these are the 3 we have discussed today, and at least I want to have solved. There is a 4th that merely has to do with global health, and that's the disease of tuberculosis. And there is a wish to reduce the number of tuberculosis patients and have very good treatments. And some of my colleagues are also very working hard in that field. And I hope that the goals of the WHO will be met I think it was the 2030 or something.
And of course, one of the ideas that you also said that intrigued me is the optical spectrometer methodology for TDM. At least that intrigued me a lot.
Yes, but I also know that there's a long way to go, because we now have new European legislation, the IVDR. Perhaps you've heard about it, perhaps not. It makes it difficult to develop these new ways of doing therapeutic drug monitoring, because there's much more paperwork, it comes close to the direction of the development of a new drug. And it a little bit limits your
It comes in the direction of developing a new drug. Although it's a technique.
Although it's a new analytical technique, the requirements are becoming the same as the development of a drug.
Okay. So the regulations are becoming more and more strict, and therefore it's becoming a longer process. Okay. Yeah. Understood. Yeah. Or process that takes more money.
So we need also more development into the regulations and to knowing how to regulate stuff probably more smoothly.
Exactly. Exactly. But this was a wonderful conversation.
A lot of topics covered. I think that a lot of people, a lot of pharmacy students will be inspired by your research.
Exactly.
Thank you for your time, and thank you for sharing this with us.
Okay, it was also my pleasure.
Thank you.
Thank you.